This video highlights research of modeling protein motions with robotics-inspired probabilistic search methods. This presentation is a highlight of research conducted in the Shehu Computational Biology Lab at George Mason University. This research is based on published work from the Computational Structural Biology Workshop in 2012 and a BMC Structural Biology Journal article from 2013. Many proteins tune their biological function by transitioning between different three-dimensional shapes. This slide shows three states of commodulin. Each of these protein structures is comprised of the same amino acid sequence and represents a system with several thermodynamically stable states. This protein can therefore act as a molecular machine, changing its three-dimensional shape to carry out different biological functions. Modeling the transitions between these states is challenging due to the large number of degrees of freedom. A family of algorithms from the robotics community, known as path planners, have been used to study this problem. Let's review the goal of these algorithms and how they apply to modeling protein systems. Path planning algorithms find a path from a start configuration to a goal configuration while avoiding obstacles, shown here in blue. In protein systems, higher energy states will act as our obstacles. This picture illustrates a very simplified problem with 2 degrees of freedom. The high energy areas are shown in red. This video illustrates the process of navigating an energetically feasible path between the start and goal states using a path planning algorithm known as RRT. The algorithm works as follows. A new point is sampled at random. The nearest node in the tree is then located, and an attempt is made to extend the node towards the sample. If the new location is lower than energy, the move is accepted. If the move is higher than energy, then the Metropolis criterion is used to determine if it is accepted or rejected. Our work extends this algorithm to work on protein systems. Since even coarse-grained protein representations consist of hundreds of degrees of freedom, randomly sampling conformations in order to extend the search tree is not practical. The process of expanding a node employs the molecular fragment replacement technique. This technique greatly simplifies the search space by utilizing a database of backbone angle vectors custom built for our protein system. A new candidate conformation is formed by performing a single fragment replacement on the parent. The energetic credibility of each candidate is evaluated using a potential energy function and the Metropolis criterion. Our method creates M of these candidates. The RMSD between each candidate and the goal structure is then calculated. The candidate that moves us closest to the goal structure is retained and inserted into the tree. A projection layer is employed to bias the growth of the tree. Each conformation is mapped into a one-dimensional grid based on its RMSD to the goal state. Selecting a node for expansion is a two-step process. First, a bin is selected, and then a conformation from within the bin is chosen. We investigate different methods for selecting the bins. Some are greedy and pick the nodes closest to the goal more frequently. A slightly softer bias towards the goal has been shown to yield a good balance of exploration, shown in the breadth of the search tree, and depth, shown as the proximity to the goal state. This chart shows some results of employing our framework on a few representative protein systems. The different colored bars represent the different biasing techniques used to guide the growth of the search tree. The results show the RMSD distance from the closest node in our tree to the target structure. This video shows the pathway discovered by our framework for a commodulin, a protein with 144 amino acids. This video shows the pathway discovered by our framework moving from the closed to open state for adenylate kinase, a protein with 214 amino acids. This research is made possible in part by funding from the National Science Foundation.